Welcome to Biology for You. In this video, we are going to discuss restriction enzymes. In the series of videos, we have been discussing different processes in genetic engineering. Here, in order to understand the basic concept of genetic engineering, like how do we cut the target DNA, how do we patch up it with the vector, how do we clone, in order to know these things, first we need to understand restriction enzymes. So because of the restriction enzymes, that is even the these molecular cloning made us possible. So here, that is the importance of the restriction enzymes. So here we are going to discuss basically what are restriction enzymes and what is their significance, followed by restriction modification system. Are R&M systems, shortly R&M systems. So third one is nomenclature. How do we name them? Fourth one is mechanism. That means how restriction enzymes cut the DNA. And fifth one is types of cutting, and followed by types of restriction enzymes. So these are the points we are going to discuss in this video. So here coming to the first point, what are restriction enzymes and what is their significance? So coming to that point, generally these restriction enzymes, the name indicates these are enzymes, and here the full name of restriction enzymes is restriction endonucleases. That means these are type of endonucleases. Nuclease is the enzyme which cuts the nucleic acid, that means DNA. So generally nucleases cuts the DNA. And this endonuclease means if you there are two types of nucleases. If you take a DNA, if a nucleus is cutting and degrading the DNA from the end portion, then it is called as exonuclease. If an enzyme, nuclease enzyme, is able to cut the DNA from the middle, then it is called as endonuclease. So these restriction enzymes are types of endonucleases. And also they won't cut DNA randomly, but they do follow some specific order. If you treat the restriction enzyme with DNA, if you treat DNA with a restriction enzyme, it will cut at very specific portions, not randomly here and there. So these specific portions is having a recognition sequence or restriction sequence for that particular restriction enzyme. So here, if you treat a DNA with restriction enzyme, it specifically cut the DNA at specific sites, that is called recognition site. So for every restriction enzyme there will be a recognition site. It will go and bind with the DNA and look for the recognition site. That means a specific sequence of the DNA. So at this specific sequence of the DNA, it made a cut. So that is what restriction enzyme is. So it is having a recognition site at which only the restriction enzyme cut the DNA, and these are basically endonucleases. That means they cut DNA from the middle. So that is it. And what is the significance of restriction enzymes? That means here basically in biological system, these restriction enzymes most widely present in different bacteria. There are thousands of restriction enzymes, and here these are part of the defense system of the bacteria. That means because of the restriction enzymes, presence of the restriction enzyme suspended in its cytoplasm, it fights against the foreign DNA. That means generally there are some viruses which attacks the bacteria. So these are called as bacteriophages. So bacteriophage is the virus that attack the bacteria. So the structure of bacteriophage is similar to this. It is having one protein coat. This all external part is the protein coat, and internally it is having DNA or RNA, any nucleic acid. So here generally these bacteriophages will adsorb on the surface of the bacteria and inject once after infection process is carried, and it will inject their nucleic acid into the bacterial cell. So here this DNA will be injected into the bacterial cell, and it will only cause infection and do the remaining work. So here this phase DNA will hijack the enzyme machinery. Enzymes of the bacteria, so that these enzymes will work for the viral DNA instead of the bacterial DNA. It will replicate the viral DNA, it will go for gene expression to produce different viral proteins, these coat proteins. So again this viral DNA will be replicated and inserted into this whole viral particles, and once after all this process got finished, it will produce multiple copies of the viral itself in the bacteria. Then the bacterial cell wall will be bursted out to release the daughter bacteriophages out. So that is the infection process of the bacteriophage. So here these bacteria must have some defense against similar this infection, right? So due to the presence of restriction enzymes, so here bacteria having restriction enzymes in its pocket. That means to fight with the bacteriophage, it have restriction enzymes. These restriction enzymes will go and bind with this bacteriophage DNA and degrade it before it will do multiplications and all. So here these restriction enzymes are able to cleave the foreign DNA embedded in the bacterial cell. So that is the basic biological significance of the restriction enzymes. And if an restriction enzyme able to cut the DNA at specific sites, not randomly, that means it is quite helpful to make fragments. That means in molecular biology processes, we need to cut the DNA into processable fragments. That means the genomic DNA or any DNA will be very large in size. If you do something with the DNA, you need to go for cutting. That means the processable small sized fragment should come, so that we can take one of the fragment which need out of the all remaining fragments. So for that we use restriction enzymes. These restriction enzymes has specific activity. That means they specifically cut at certain positions. So these are also used in the cloning process. That means if you want to cut the target DNA, if you want to cut the vector as well, if you use the same enzyme to cut the target DNA as well as the vector, so both will have same kinds of cleavage products. So they will be just easily going and attach with this target DNA will be easily inserted into the vector. So that is the major experimental significance of the restriction enzymes. That means they produce recombinant products. So for making a recombinant product we need to have restriction enzymes. For doing restriction digestion, to make small fragments out of the DNA we need to have restriction enzymes. That is the basic experimental or molecular biological significance of the restriction enzymes. So coming to the next point, restriction modification systems. What exactly restriction modification systems? Okay, let us again take this bacteria example. The bacteria which has its xenomic DNA, and also it has newly invaded foreign DNA. So this may be a phase DNA. That means this phase able to inject its DNA into the bacterial cell. What will happen? These restriction enzymes will be active, and these restriction enzymes will cut the foreign DNA so that it will be no more active and it cannot hijack the host enzyme machinery. So that is how the foreign DNA will be eliminated. So what if these restriction enzymes are also active towards the genomic DNA, right? So they may cleave this genomic DNA also, but which is not the actual case, because these restriction enzymes are not active towards its own DNA. So the bacterial own DNA will be protected from this restriction digestion. So due to the presence of methyl groups that are added here and there, once after the bacterial replication got finished, these methyl groups will go and attach with the newly synthesized bacterial DNA during its replication itself. So these methyl groups protect these restriction sites and protect the DNA from the digestion by its own enzymes. So that is called modification. So here bacterial own DNA is modified in such a way that it will be protected from its own restriction enzymes. But the foreign DNA, which is not having this modification system, restriction modification system, it will be easily degraded. So that is what restriction modification system is. So coming to the next point, that is nomenclature. So here how do we name the restriction enzymes? So there are set of rules called, in year 1973 scientists called Smith and Nathans made some set of rules for us to nomenclature the restriction enzymes. That means there are many thousands of restriction enzymes all around, and among which some handful, that is up to some hundreds, 400 and enzymes are in experimental significance. They are most widely studied. So among these enzymes we need to know what is what, which cuts at which point of the DNA and how it works, which kind of which type of the restriction enzyme it is. So in order to identify everything, every restriction enzyme, we need to give them particular names. So here these rules of nomenclature were made by these scientists, and here we have rules like: first, we need to give a restriction enzyme with 3-letter acronym. That means there must be at least 3 letters. Okay, let us consider a popular restriction enzyme to understand what are the nomenclature rules. Here HIND. So HIND3 is one of the popular restriction enzyme. So how do we give name to the HIND3? So here H stand for the first letter of the genus, that is Haemophilus. So that is the first letter of the genus, and IN stand for first 2 letters of the species. This is from influenza. That means H stands for Haemophilus, I stands for influenza, and D indicates the serotype. So this is the variety or serotype of the bacteria from which this enzyme got, HIND3 got isolated. So what is this 3? Roman letter 3 indicates. This 3 indicates the number of the restriction enzyme in a sequence that is isolated from this particular strain of the bacteria. That means there might be 3 enzymes. And this is the 3rd enzyme which got identified in the Haemophilus influenza serotype D. So that is how we give names to the restriction enzymes. Coming to the next point, how restriction enzymes works. That means what is the mechanism of the restriction digestion. So as we already discussed, they made cuts in the DNA, but how do they act as molecular scissors to cut the DNA? Basically these are hydrolyze the DNA. So the process hydrolysis, nothing but hydrolysis. Okay, let us consider this as the DNA to be digested with restriction enzymes, and we treated it with restriction enzyme. These restriction enzymes are randomly attacked on the DNA. They go and bind with the DNA at random sites wherever they could be. So these restriction enzymes will go and bind with the DNA at random sites, and then they look for the recognition site on the DNA. That means these restriction enzymes go and bind with the backbone of the DNA. So they'll go and bind with the backbone of the DNA, and they slide forward, and once after they recognize these specific restriction sites, that means a specific sequence of the DNA. So once after they reach the specific sequence of the DNA, they'll stop and cut. So here these restriction enzymes will be activated once after they reach the recognition site, and once after they got activated they made cuts. So here, as we already discussed, the process is nothing but hydrolysis. Okay, let us draw structure of the DNA. So this is the structure of the DNA. That means here is a base and here is the other base, and let us consider it as the recognition site where the restriction enzyme identify the DNA to cut. So this is the phosphodiester bond, and generally this phosphodiester bond is hydrolyzed by the restriction enzyme. Here this restriction enzyme is having active site. That means any enzyme will have active site. The responsibility of active site is for its function. That means here this restriction enzyme having aspartate molecule in its active site which contains Mg2 plus ions. So here these Mg2 plus ions present in the active site are responsible for the cutting. So here Mg2 plus ions holds the water molecule. So they hold the water molecule, and these Mg2 plus ions made the water molecule to be polarized so that deprotonation will be easy for the enzyme. So they will donate these molecules, water molecules, to the phosphodiester bond, and they will cut eventually. So for cutting there is another magnesium ion. For this hydrolysis process there will be another ion. So here generally restriction enzymes go and bind with this skeleton of the DNA, and also they either may form once after this nucleophile attack has been carried, they may form intermediate, or they may directly cut without making an intermediate structure with this DNA. So they'll go and bind to form intermediate or they may not form intermediate product and directly cut the DNA. So here this water molecule is donated to the phosphodiester bond. So this phosphoryl transfer to the water, we can say. So here this will be cut into two fragments. Here will be base, it will be oxygen, and so this is 5 prime, right, and this is 3 prime. So here this OH group will remain with the 3 prime end, and here 5 prime will be taken the phosphate group. So that is how the So they will be separated as 3 prime and 5 prime, new 5 prime ends. So that is how the mechanism of restriction enzyme is. That means it makes the hydrolysis of the phosphodiester bond. These Mz2 plus ions which are present at the active site made it possible. That means they donate the water molecule, they hold the water molecule and make it depolarized and deprotonated. So that is the mechanism of the restriction enzymes. Coming to the next, types of cutting. So generally restriction enzyme, let us consider it as a DNA. If restriction enzyme able to cut at the same point in the two strands of the DNA, okay, at the same nucleotide position, then the cuts will be blunt ended. Okay, so it will be at same nucleotide position, so the cut is called blunt ended. What if a restriction enzyme able to cut the DNA at different positions? Say for example the cutting is like this, then the product will be similar to this. The nucleotide positions are different, so what we can say, these are sticky ends or staggered ends. So there are two types of restriction enzymes again which can form blunt ends and the staggered ends. Let us go with the examples. So here for blunt end cutters, we can say examples like LU1, SMA, HIND, HA, etc. These are the restriction enzymes makes the blunt ends. Coming to the sticky ends, sticky end cutters. These sticky end cutters or staggered end producers are BAM, H1, Eco R1, there are many blunt end cutters and sticky end cutters as well. So these are the main examples of the blunt and sticky end cutters. So we already discussed types of cutting, and coming to the last point, types of restriction enzymes itself. So basically there are 4 types of restriction enzymes. Restriction enzymes type 1, type 2, type 3 and type 4, and based on their mechanism, structure, the cofactors they contain and the mechanism, mostly mechanism will be quite similar, but the activities they possess, these makes the differences in the different types of the restriction enzymes. So these 1 and 3 are much similar, so sometimes we can consider it as same unit itself, and this restriction enzyme 1 and 3 are the similar. And coming to 2, restriction enzyme type 2 are most having biological significance. Generally this restriction enzyme 1 and 3 are part of restriction modification systems. That means they possess methylase activity and the restriction activity also. And these restriction type 2 enzymes have only restriction enzyme activity. That means they'll cut the DNA. They don't have the activity to add on the methyl groups to the DNA. Coming to the fourth, this type 4 restriction enzymes are basically cut the DNA only when the DNA is already got methylated. So these are the main differences of the restriction enzymes, different restriction enzymes. So basically type 3 and 4 enzymes are not very much significant. So here generally we use type 1 and type 2 enzymes. So let us discuss details about the type 1 restriction enzymes and type 2 restriction enzymes. So here type 1 enzymes are restriction enzymes as well as methylases, as we already discussed. These are only restriction enzymes. They are able to cut the DNA only, and these enzymes are multi-subunit enzymes. That means they have one subunit for the recognition of the DNA, recognition of the restriction site. The one subunit is for the cutting, restriction cut, and the other subunit, the third subunit, is for the methylation activity. So there are three subunits. Coming to the type 2 restriction enzymes, these are homodimers. Dimers with restriction activity. Coming to the next point, these enzymes, as we already discussed, having restriction enzyme activity and methylases activity. That means they must have different cofactors accordingly. That means they have SAM cofactors and Mg2 plus ATP. So this SAM is nothing but S-adenosine methionine. So this SAM is the major donor for the methyl group, as it is having methyl group in it. This cofactor donates the methyl group to the DNA. So because of the presence of SAM, the type 1 restriction enzymes are able to donate methyl groups, or shortly they act as restriction methylases, and this ATP is the donor of the energy, as we already discussed. Mg2 plus is the cofactor for restriction digestion. So coming to the type 2 restriction enzymes, the cofactors are majorly Mg2 plus ions. That means they only do restriction cutting. So Mg2 plus ion is enough. Coming to the fourth point. Generally, these type 1 restriction enzymes made cut, they'll recognize recognition site. That means they identify the sequence of the DNA which is specific for the cutting, and that is called recognition site. They identify the recognition site, but they will not cut within the site. They will not cut the DNA within the site, but it is far away from the recognition site. That means it's like some thousands of base pairs away from the recognition site, they will cut the DNA. So that is the other DNA. They cut the DNA away from the recognition site. And here, coming to the type 2 restriction enzymes, they cut the DNA within the vicinity of the recognition site. That means if they recognizes the restriction site over here, then the cutting will also takes place in the same place. That means within the recognition site, they will cut the DNA within the vicinity, or we can simply say recognition site. So the examples for these type 1 restriction enzymes are ECO, and the examples for the restriction enzyme. Type 2 are, there are hues like Hind3, Hind2, Eco R1 and SMA1, ALU. So these type 2 restriction enzymes only have wide applications in molecular biology experiments. So most of the enzymes we found in the laboratory are restriction type 2 enzymes. So that is what types of restriction enzymes. So here in this video we discussed what exactly are the restriction enzymes and what is their significance, their molecular biological significance as well as the biological significance. And we discussed R&M systems, that is restriction and modification systems, and we were going through this nomenclature, how do we name the restriction enzymes, and followed by the what are the mechanism of the restriction enzyme action, that means how do they cut the DNA, followed by different types of cutting, and lastly we discussed different types of restriction enzymes. So by this I can expect somebody will get the basic idea about the restriction enzymes and how do they are helpful for us in molecular biology experiment, and what are their importance in the bacteria in which they majorly present. So that is what the restriction enzymes are. So thanks for watching